Okay, good. So, welcome to everybody. I am Joris van der Putten, the president of IABS, International Alliance for Biological Standardization. And I thank all of you for participating in this other webinar on COVID and a particular important subject on human infection studies. You know, IABS' role is to convene and to bring all stakeholders together, and that's what we are doing again this afternoon. I am so proud of so many participants of all over the world to discuss, pose questions on a very hot topic, I would say, a very important one, and probably an important one in the fight against the COVID-19. I want to thank particularly our co-sponsors, HVivo, the Wellcome Trust, and HICVAC, and particularly all the speakers and the session chairs and the participants on the round table. As you can see, all participants will be invited to or will be on mute. Questions are posed in the Q&A button, and I will do my best after each session then to try to convey all the questions to the speakers and the panelists. And so as said, this will be discussed after the series of presentations. And if there are many questions, given the time is limited, I apologize already if not all questions cannot be answers. And please take a few minutes at the end of the webinar in order to answer the survey. I pass it over now to Peter and then to Isabelle and Jean-Hugues. Peter.
Yes, thank you, Joris. Just to welcome everybody as the chair of the Human Vaccine Committee of IABS. I've been highly involved in the organization of the writing of this document. And as you can hear, I say organization of the writing. I have to thank a lot of people that worked very hard on the production of this document. And as you can imagine, this was not an easy going, but we made it, and we made it in time. So thanks for all of you that have participated with comments in this document. All of you received the document. Those that registered last minute have received it half an hour ago. So everybody has the document, and we want to have a fruitful discussion on this document. We have two co-chairs of this afternoon, Jean-Hugues Trouvin, who is the former president of the Biotech Working Party at EMA, and we have Isabelle Beckeridging-Ding from the Polelig Institute, who is head of microbiological service. I give the floor to Jean-Hugues, and we can start. Over.
Thank you, Peter. So I will chair this first part of this webinar, which will be dedicated to 5 or 6 presentations to illustrate the development of this white paper on the development and manufacturing condition for a challenge agent. For that, I would like to give the floor first to Karine La, from HVivo, Associate Director for Clinical Science, and she will present briefly this document. After that, we will have a presentation, 10 minutes each, and if possible, keep your question for the roundtable, of course. Karine?
Yeah, thank you, Jean-Hugues. Well, good afternoon, everyone. My name is Karine La from HVivo, which is a service company specialized in human infection model studies. And first of all, thank you to all the IABS team for the organization of this webinar. And before we start, I would like to give you an overview of this project and its purpose. As you may know, human infection studies, also known under other names, are more and more used for, these past few years to evaluate vaccines, prophylactics, and treatments, but also to have a better understanding of different diseases. And these studies aim to expose a small number of individuals to pathogens, which are also called challenge agents. And the big question here is, how can we produce a challenge agent in such a way we have at the end a high quality and safe product to be inoculated into humans? And so that is why recently different experts raised the increasing need to establish quality principles for the development and for the manufacturing of challenge agents, considering that currently challenge agents are classified differently by regulatory bodies in different parts of the world. And this is why, so HICVAC, which is an international network of researchers who are developing human infection studies, and the Wellcome Trusts have decided to undertake this project that aims to develop a white paper where a guidance support primarily for researchers is proposed. And this white paper will include low and medium income countries considerations, but also how challenge agents can be produced outside a GMP accredited environment. And so for the development of this white paper, a consortium was set up last year to deliver this project with HVIVO as a lead coordinator. And then the consortium has members coming from different regions and with expertise on different types of pathogens, bacteria, viruses, and parasites, but also expertise in regulatory and CMC. Of course, to make it more consistent, we take into account LMIC considerations by consulting people coming from this region, and also a large panel of experts in CMC and regulatory. So today, the consortium is happy to release this first draft of the white paper and to give a presentation on the different key sections of it, which will focus in priority on fundamental quality principles to develop and manufacture challenge agents, and also how we can formalize this development and manufacturing. And after this webinar, we will refine the document following feedback from the audience of this webinar, and we will consult also more experts to finalize it. And then we expect to have the final version by, let's say, mid-August 2021. This is all for me. I hope that gives you a big overview of the project. Happy to be part of it.
Thank you, Karine, for this introduction to this project, and the document will also be available for all participants, of course, and even it has already been made available. The second presentation will be by Hilde de Prater from European Vaccine Initiative. She is a CMC expert, and she will give some general consideration essentially on the quality development. Hilde, floor is yours.
Thank you, Jean-Hugues, and thank you also for the organizers to give me the opportunity to be part of this project. Okay, so I hope you can see the slides. So I will give a little update on the general considerations that we took into account when we were writing this white paper. And so the general considerations are related, of course, to the designing and producing of the challenge agents. If we can go to the next slide. So there are some key principles that we followed or that we took into account when starting working on this document. And so one of the major principles that we have to keep in mind is that despite of this white paper, that we always have to look at the applicable local, national and regional guidelines that might be available on the manufacture of challenge agents, and that we should always consider them where available. A second key principle is that we have to think about the intended usage of the agent and the safety of the volunteers participating in the trials. Yeah. And then a third point that we take that we can mention here, and this is especially for the many scientists that are developing challenge agents, is the guidelines that we took into consideration to write this white paper. And so here we mainly focused on ICH guidelines, as they are quite often adopted worldwide. And so for this particular case, for the development of challenge agents, a challenge agent, I think we can consider as biological products. Yeah, and so we took into account the ICH Q5A on viral safety evaluation, the ICH Q5C on quality of biotechnological products, also considering stability testing, the ICH Q5D on derivation and characterization of cell substrates used for the production of biotechnological projects and biological products, the ICH Q6B on specifications and test procedures and acceptance criteria for biological products. In addition to those guidelines, we also took into consideration some principles and some wording from the ICH Q7 guideline on GMP manufacturing, although that this might not always be applicable to all types of challenge agents. We took into account ICH Q8 on pharmaceutical development, Q9 on quality risk management, Q10 on pharmaceutical quality systems, and Q11 on development and manufacturing of drug substances. So in all of these guidelines, there were items that are interesting to consider when developing new challenge agents. If we go to the next slide. So what is the process that we propose to follow when considering developing a new challenge agent? There are different steps that we propose to follow. And the first step is what we call characterization, or the development of a target product profile. For those not familiar with the CMC and regulatory nomenclature, a target product profile is designing a profile of your challenge agent to identify the key characteristics of the challenge agent and its uses. So what you have in mind that how the challenge agents should look like. And so if I can make a more, how to say it, a less abstract definition of it, then I would say, look at the fact if you would like to design a new car. Yeah, and then if you would like to design a new car, also there you could say I will develop a target product profile, and this could then include items like I want my car to look like this, I want my car to have a performance like this, I want it to be an environmental friendly car, and I want it to be a safe car, for example. Yeah. In the next step, once you have developed your target product profile, you will go one step further and you will think about the quality considerations of your challenge agent. And this can be worked out in a quality target profile. So this is basically to translate the characteristics from the TPP into a quality considerations to ensure the safety, the potency, and the stability of your challenge agent. Again, when we make the reference to a car, this would be then, for example, if you say, I want a performant car, so this means that you will have to think about the quality principles for the motor that you will use in your car. The same for an environmental aspects. Again, this could be linked to the motor of your car. You want to have an electrical motor, etc. If you think about safety aspects for the car, it would relate to, for example, the brakes, the safety belts, the lights, and all the items related to the safety aspects of your car. In the next step, Based on the quality target profile, we will define what we call critical quality attributes. And so this is, without going into detail for one pathogen in particular, important quality attributes will always be related to the safety and to the pathogenicity of your challenge agent. And it will always include the following items. It's about the identity of the challenge agent, the potency, the purity, the stability, and this could be genomic stability of the challenge agent, the dosage form, sterility, and transmission. And in the next step, we will go then to the quality control strategy. And this is then the strategy that should be viewed as a set of processes and testing to be put in place to ensure that the manufacturing plan is followed and documented. And a control strategy for a challenge agent includes elements which impact both the process used and the product. And if we make the reference again to the example with the car, so also for a car, before you can drive the car, your car will need to be tested and need to be compliant with certain criteria before you can drive it. So if we go to the next slide, we will focus now a little bit more on the challenge agent topics. And so if we come back to the target product profile, so which items can be covered in a target product profile for a challenge agent? So this will not only be focusing on quality aspects, but this will be more general. In a target product profile, you will consider some non-clinical aspects. This could be about the mechanism of action and the potency. More specific, it could be related to the virulence factors of your challenge agent, the pathogenicity of your challenge agents, what do you already know? Non-clinical aspects can also be related to, do we know something about the sensitivity to therapy? Do we know something about the impurities? So when we will prepare the challenge agent, and do we already know that there could be contamination of other pathogens present in, for example, the isolate of the primary challenge agent? Other aspects to be considered are clinical aspects. So this could be related to the dose regimen. So you might have to do some dose studies to determine what would be the optimal dose for your challenge agent to be used. Yeah? It could be linked to the administration route. It will be linked to safety aspects. So you will have to determine for each challenge agent what is the acceptable adverse event profile that I can accept for this agent. And it will be linked to efficacy and potency. So what we want to see is a challenge agent that is safe, but at the same time is able preferentially to create a natural disease in the volunteers. Other aspects to be taken into account are regulatory aspects with regard to registration processes once you want to use the challenge agent. And then, of course, also biosafety considerations, because it might be necessary to work under BSL-2, BSL-3 environment, and quite often an environmental risk assessment might be needed. And then last but not least is when you design a challenge agent is to think about some of the CMC aspects. And this is then related to the presentation of the challenge agent. Will it be a liquid? Will it be freeze-dried, lyophilized? What about the formulation aspects? We have to think about stability and storage. And here, I mean stability. We would like to put a shelf life on the challenge agent, but also we have to think about in-use stability for the challenge agent. And then we have to think about all the items related to production of the challenge agent. Can we have an affordable production process? Will it be able to be scaled up, and so on? So once we have this TPP, if we go to the next slide, we want to translate it further in a quality target profile. And so here, for example, if we think in for the quality profile, there will be items again related to the dosage form, to the route of administration. For example, is it the oral route, intravenous? Is it via an insect bite, and so on? So quite often in the route of administration for some challenge agents, we might use vectors, and this might have an impact, for example, on transmission of other vector-borne diseases. And these are things that we have to take into account when we think about the quality profile. There is the stability, and as I mentioned before, this will be stability during manufacturing process, but also storage over time. This will be linked to real-time stability studies and also in-use stability studies. For example, we can think here about what could be important in a production process is the number of passages that is needed to produce your challenge agent, because this could have an impact on, for example, genomic stability. There could be mutations. Will this have an impact? Yes or no? How we will follow up on this. Then there is a whole list of quality attributes. This can be physical attributes of the challenge agents. This will be linked to the identification of the challenge agent. This will be linked to the assays that we will use. Assays, what level of validation do we want for our assays? Will it be sufficient to have qualified assays, or do we need validated assays? We will have to think about the purity and impurity profile, about degradation products, microbial limits and advantageous agents testing, and then biological activity and potency. And then also in the end, for your quality target profile, you will think about your container closure system, because you will need a suitable container closure system to achieve the target shelf life and to ensure challenge agent integrity during shipping. So in the next slide, we give one example of an item that is translated from TPP to QTPP. So for example, if we have in the target product profile, when we think about the potency, in the target product profile, this might be described as the need to confirm and validate virulence factors, if the information is available, of course, and causing generalizable asymptomatic infection carriers or symptomatic disease. When we translate this into a QTTP, the challenge agent quality attributes, there it will be in the item biological activity potency. And then the potency of the challenge agent should be evaluated. And where needed, we have to think about reference materials to be developed to ensure consistency in testing of different challenge agent batches. If we go to the next slide, so once we have the QTTP and have identified potential quality attributes, a next step will be to list all these potential quality attributes and to do a risk assessment and prioritization of these quality attributes. And when we do this exercise, and the result of this exercise should be that we identify what we call the critical quality attributes. And the critical quality attributes will be those items that we will test during either the manufacturing process or at the end that we will test on the challenge agent itself. So this will lead to in-process testing during manufacturing, to characterization testing, and then at the end, once you have your challenge agent produced, this will result in definition of the quality control testing, QC release testing that you will do on your challenge agent. So this is a top line, a little bit the process that we would propose to follow when designing a new challenge agent. And so if we go to the next slide, I just summarized the main steps once again. So it's a stepwise process. Yeah, it starts with the establishment of a target product profile. Then it follows with follow-up steps for the identification of the key quality attributes and release criteria. And then in the next step, the last steps, all of this will be transferred the process for further routine manufacturing. And so this might result in establishment of standard operating procedures, documentation to monitor and control the manufacturing process, and then the utilization of qualified validated methods to test the final product or some critical steps of the manufacturing process itself. So this is all for me, and in the next presentations, we will go a little bit deeper in these different steps that are determined here. Thank you.
Thank you, Hilde, for this presentation and introducing the general principles to be considered for the development of a challenge agent, considering that the quality attributes of the challenge agent is fundamental and is indeed critical for the reliability of the clinical challenge, which will be performed using this challenge agent. So the next presentation will be given by Edi Suwarnapunya from the Walter Reed Army Institute. He's a microbiologist in charge of research on infectious diseases. And he will speak about the challenge agent selection and characterization, including adventitious agent testing. So this will be a direct application of what Hilde has just presented. Edi, floor is yours.
Thank you. Can everybody hear me?
Yes, clear.
Okay, great. Yes, my name is Edi Suvarnapunya. I work in the Department of Diarrheal Disease Research at the Walter Reed Army Institute of Research. And I'll be talking about a challenge agent that we've made in our diarrheal disease work. So first thing to get out of the way, number one, this is the building where I work. This is a campus at Silver Spring in Maryland. The views expressed in this presentation are mine and not necessarily the Department of the Army or the US government. And also the mention of any trade names doesn't imply any endorsement. So you may wonder why diarrheal disease is a focus of the Department of Defense. And the reason why is because it's been determined that there is a high risk of diarrheal disease to impact operations all around the world. So on the left here, this is a map that shows that relative risk. And you can see where the darker colors represent the highest risk, but really it's a high risk around most of the world. That also correlates to a rise of Shigella sonii in particular. So a number of pathogens are diarrheal pathogens. We work on Shigella and Campylobacter and enterotoxigenic E. coli, but I'll be talking about Shigella today and specifically Shigella sonii. So you can see on the right, this is a map of showing the relative prevalence of Shigella sonii and Shigella flexneri, which are the two most prevalent serotypes in the world. And you can see here that Shigella sonii, that in the darker colors, is more prevalent than Shigella flexneri, mostly in developed countries rather than developing countries. So as the world shifts to more development, we predict that sonii will become more of a problem than so Shigella sonii we were interested in developing as a challenge agent, and historically the 53G strain has been used in clinical trials in the past. This was a strain that was isolated in 1954 from a 5-year-old child in Tokyo. that was eventually transferred to the Center for Vaccine Development at the University of Maryland. Since then, a number of trials listed here in this table have used 53G as a challenge agent to test various vaccine candidates. The target CFUs that were used in these trials are generally around 500 CFUs. These CFUs were delivered or basically prepared by making bacterial lawns on agar that were then harvested and diluted in skim milk and delivered to subjects. So over here, you can see the attack rates associated with diarrhea, dysentery, and fever. And so you can see these are generally around 50%. And all these trials were conducted in the United States with naive volunteers. In 1995, a study by Kotloff et al. Was published showing that for Shigella flexneri, bicarbonate buffer as the vehicle for delivering the agent resulted in a higher attack rate that also seemed to be more consistent. So trials since 95 have used bicarbonate buffer in any trials involving a challenge. In 98, streak plates from the CVD were obtained by Rare to manufacture a frozen glycerol master cell bank and production cell bank. And those lot numbers are shown here. Subsequently, vials of those cell banks were sent to the Armed Forces Research Institute of Medical Sciences, or AFRIMS, where they made their own frozen glycerol lot, shown here for challenge in studies that were conducted in Thailand. So two of them in 2012 and 2016 used either this frozen glycerol lot or lot 0593. And again, incorporating the bicarbonate buffer concept, rather than use the buffer as the actual vehicle for the challenge agent, this was administered separately to the subjects immediately before administering the challenge agent in water. And another small improvement from previous trials was that there was a dose finding study done where they reported the actual CFUs that were delivered rather than just the target CFUs. And they found in 2012 that 1680 CFUs gave a high attack rate for dysentery. In 2016, they published a follow-on study where they used this dose in an efficacy trial, but were unable to reproduce that high rate. So a summary so far of the issues that I've touched on. A relatively low attack rate seen with this challenge strain using the procedures that I outlined. The attack rate was inconsistent. That could be because of the naive, comparing a naive population in the United States versus endemic population in Thailand. It could also be due to the fact that we are using multiple different lots. So the earlier studies in the United States, it's not clear whether they were all from the same lot. It's probably unlikely. And then in Thailand, as I mentioned, two different lots actually were used for the two studies that I talked about. Additionally, because these bacteria need to be first streaked out on a plate for isolation, colonies need to be selected for lawns, and lawns then need to be harvested and diluted to working concentration, that introduces a lot of variability itself. And so for Shigella sonii, it's convenient in the sense that virulent colonies can be easily distinguished. So virulent colonies are also called Form 1, and those are shown here circled in green. This picture is TSA media. The red circled colonies are Form 2. These colonies have lost the virulence plasmid. And the reason why you can distinguish that by morphology is because the O antigen operon in Shigella sonii is located on the virulence plasmid. So it's an easy, quick and easy way to determine just by colony morphology. A little more sensitive way is by using Congo Red Agar. So that's shown here in this bottom picture. And in this picture, you can see that, again, virulence plasmid containing colonies show up as red. Colonies that have lost the virulence plasmid show up as white. And this is because what's also encoded on the virulence plasmid is the type 3 secretion system. And this needle structure tends to bind up the Congo red. And that's what leads to the red color in plasmid containing colonies. So when you're preparing the challenge strain, it's obviously important to select these virulent colonies. There is a certain rate of plasmid loss in Shigella sonii and Flexneri. Shigella sonii tends to have a higher rate of virulence plasmid loss. It can be up to 50%. So that's an important consideration. Also, I touched on the inconsistent administration, challenge administration procedures, whether you're talking about the vehicle that the challenge is prepared in, or the sequential use of bicarbonate followed by the challenge. So this is a visual depiction of the preparation process that I just described. Again, there are 2 passages here that are required before the inoculum is actually prepared for administration. So this is when using 53G that's prepared from glycerol stocks traditionally. So we thought that a lot of this variability could be eliminated by using 53G that is lyophilized. So rather than go through these 2 passages and introduce the opportunity for mutation or mistakes in selection of avirulent colonies, we have the lyophilized product already in a vial that just needs to be rehydrated and diluted. So these first 2 days, you can hit the beach using the lyophilized preparation rather than the traditional plate scraping preparation. This is the product that we made, again, lyophilized, lot 1794. This was manufactured in 2013. Contains about 4 × 10⁹ CFUs per vial in a 2 ml volume. About 80% or greater than 80% Form 1 CFUs are present in this lot, and it's stored at -80. The preparation for this type of lyophilized bacteria is simply to transfer the vial from storage to ice, let it equilibrate for 30 minutes, then the lyophilized bacteria are rehydrated with water, an equal volume of water, swirled, and then diluted to the desired concentration. This is a overview of the manufacturing process that we used. From a single research seed vial, we create a master cell bank and a production cell bank, which are identical except they're one passage removed. From the production cell bank, we create a fermentation seed culture, which is just a shake flask, as are these. That starter culture is used to inoculate a 30-liter fermenter, and then vials are filled, and those the product in those vials are lyophilized and released after a series of tests. So this is just depicting that flow, as well as the number of vials that you get typically in a master cell bank, a production cell bank, and then the final product lot of about 1,000 vials divided into vials for use, as well as for long-term studies and testing. So again, starting with the research seed, we pick virulent colonies to make a master cell bank and a production cell bank. Because these are shake flasks, the testing is really quite basic. It's just a gram stain of purity of streak plates by visual inspection of colonies, the type and appearance of the colonies, biochemical identification, and antibiotic susceptibility testing. The creation of the fermentation seed culture, again, is almost identical. It involves picking virulent colonies and starting the culture. Colony morphology and viability is assessed on this culture. And I should point out that this, so going back here briefly, this from here to here happens within sort of a half a day. So it's not like the testing that we do allows us to stop and adjust. It's really retrospective on the process. So the fermenter culture is also tested. A sample is withdrawn, and these are plated out and tested as shown. The lyophilized, the cryopreserved bulk product before vial filling is also, a sample is withdrawn and tested as shown. The lot release involves these specific tests. So just a visual examination of the lyophilized product in the vial, pH, moisture content, viability or total number of CFUs, as well as the percent Form 1. So these are the specifications. We do expect to exceed these specifications, and we do. So a little bit about Shigella pathogenesis, so you understand the subsequent tests. Shigella, as I mentioned, has a type 3 secretion that allows it to invade epithelial cells. Once inside the cells, Shigella can spread to adjacent cells by nucleation of actin that involves VIR-G. So you can see that in a plaque type assay where you infect epithelial monolayers with virulent Shigella, and they make plaques over time. If you don't have VIR-G, the monolayer stays intact. And then this invasiveness and intercellular spread is shown in the guinea pig keratoconjunctivitis model. So invasion alone leads to a normal guinea pig eye. But if the strain is able to spread intercellularly, the infection spreads throughout the eye, and you get purulent keratoconjunctivitis. So post-lot release testing can be broken down into two categories, basically, purity and pathogenesis. Purity, again, involves biochemical ID, antibiotic susceptibility, mitomycin C induction of lytic phage, genome sequencing, and the microbial limits test, or MLT. The pathogenesis testing involves looking for surface expression of IPA-B, which is a surrogate for containing the virulence plasmid, invasion, plaque formation, and virulence, as I discussed. The microbial limits test, I'm sure you're all familiar, but these are the specific agents that are defined in the USP version of the microbial limits test. In particular to the 53G lot that we manufactured, we also tested for these two agents because these were manufactured 6 months prior to the manufacture of 53G. Long-term studies, this is an example of one of them. It's a forced degradation study. So normally we store the product at -80 degrees Celsius. So we wanted to see the effect of storage at off temperatures. As you can see, 37 degrees over 10 weeks, there's about a 4-log drop, 5-log drop. Routine testing that we do on the lot, we're on an annual schedule now, and that really just involves assessment of total CFUs as well as Form 1 CFUs. So you can see that we're above 80% over 8 years of testing, and the total CFUs has remained relatively consistent. We can, of course, do a number of tests, and we do ad hoc testing as necessary, but this is just a reminder that we can do, whatever tests we want to do, but keeping in mind that we have a finite size of our product line. So I talked about 53G. We do have Flex 2A glycerol stocks available for the traditional plate scraping method. This is 2457T is the strain name. And then later this year, we have scheduled to make a Shigella flexneri 3A strain, J17B, in a similar manner that we use to make the 53G strain as lyophilized vials. And then I'll end the talk here by going back to this diagram showing the relationship of TPP to QTPP and CQA. All of the tests that I've talked about and the considerations that we've made during the manufacturing process fall into one or more of these categories. And we certainly didn't have a structured formal framework such as this. So I look forward to the discussion of the group as to how we think about this and where we classify this and how to make use of this going forward. Thanks.
Eddie, can you shorten a little bit because we are running over time? Thank you.
Yes, I just ended.
Thank you, Eddie, for this presentation. That's a very nice illustration, in fact, of the need for a relevant quality target product profile. I agree with you. Maybe not all the criteria should be taken into account, but you have clearly illustrated that you need to identify the limited factors for the reliability of your challenge agent. But we will discuss that certainly during the roundtable. So the next presentation will be given by Alan Bell from hVivo. He's a Director of Translational Operations, Respiratory and Infectious Disease, and he will speak about manufacturing and control. This will certainly be a new application of what has already been presented by Hilde. Alan, floor is yours.
Sure, thank you very much. Thanks to everybody for their attendance today. I've been asked to give a brief overview of the manufacturing and control section of the document. So the manufacturing and control process encompasses the definition of what the challenge agent characteristics are, the definition of the quality considerations to ensure safety, potency, and stability of the challenge agent, a definition of a quality control strategy, as well as the routine manufacturing itself. In terms of the quality control strategy, this strategy is to look at the processes and testing that need to be put into place to ensure the manufacturing plan is followed and documented. The control strategy for a challenge agent includes elements which impact both the process itself as well as the product. And its purpose is to overall look at how we meet the acceptance criteria during the manufacturing process. On the right-hand side, you'll see a list of a number of key elements to consider. These include the description of the acceptance criteria for QC release, the manufacturing process itself, test controls and what are recommended to be implemented, considerations around formulation, stability, and finally labeling. And it's these key elements that we're going to cover over the next few slides. So starting off with the manufacturing process as a whole, some key elements to consider is that it should include a summary description of the whole process. One example of demonstrating this is through the use of a flow chart, as you see in front of you. We also want to be able to identify and highlight critical steps. And the process description itself should help identify any tests that are to be implemented and any acceptance criteria applied. We should give some special attention to biodegradable materials and environmental changes, such as changes in pH. Also, descriptions around the procedures used in the transport of the material during the manufacturing process, as well as the storage conditions. If and where possible, the manufacturing area itself should be physically separated from the procurement area to try and reduce the risk of cross-contamination. And we should finally also apply some attention to the processes around the destruction of the challenge agent. In terms of formulation, whether this can be a fresh formulation, lyophilized, cryopreserved, or frozen, it's important to consider and apply attention to the lyophilized and the cryopreserved and frozen, as they may be able to produce more reproducible challenge agent potency. But the process should be carefully assessed because it can have an impact on the challenge agent quality during the manufacturing process. Test controls are used to verify that the process and procedural controls are performed as expected. One form of test control is in-process testing. In-process testing is usually some form of analytical test or functional test to ensure that selected manufacturing operations are performing appropriately to achieve the intended quality. And you see before you a list of the typical analytical performance characteristics utilized in this process. In terms of release testing, the results should be documented in some form of a certificate of analysis. And this certificate should include identification, biological activity, such as potency and purity, quantity, and microbiological quality testing where necessary. And this includes adventitious agent testing. In terms of characterization, this is for testing that's applied outside of the lot release, and it's looking mainly at comparability. It's also looking at the confirmation of genome stability after passages, as well as potency, and to check that there has been no attenuation of the strain. These processes themselves can then be overseen by process monitoring. So process monitoring should be implemented when there are several batches of the same challenge agent being produced on a regular basis. Process monitoring means using selected attributes and parameters in order to assess quality and be able to trend it over time, in order to enhance the confidence in the normal distribution of the attributes being measured. The frequency in which the process monitoring occurs should be reviewed and adjusted based on the trends and the data outputs. And we should possibly include limits for evaluating those trends. In terms of stability, a stability plan should be generated that assesses the stability profile of the challenge agent during storage. This should include a stability protocol, a proportion of vials that are available for stability testing, as well as real-time studies or stress-type studies, such as PCR to assess genomic stability. The stability plan should also have tests that are justified and qualified. The test should be conducted on the dosage form that's packaged for the clinical trial use itself. And the manufacturer must prepare a stability plan for each dose level to monitor the stability over time. A key point to just draw out from this is that when relevant and where relevant, genomic and phenotypic stability should be assessed during the manufacturing and the storage phases, as well as other criteria attributes such as viability or motility, and in certain instances, such as the example here, schistosome, also morphology. And then finally, concerning labeling, the WHO good manufacturing practices for biological products should be followed as much as possible and where possible. The label itself or the leaflet that accompanies the vial should as a minimum include the name and expiry or retest date of the challenge agent, the concentration, as well as the number of doses if the product is being issued in a multiple dose container. In terms of distribution and transport, any temperature recommendations should be clearly provided. And that's for temperature during storage as well as transport. The shipments themselves should be maintained within a specified temperature range. And where required, packages should contain cold chain monitors. And overall, the packaging process for shipment should conform to the IATA standards. And finally, one key point in this area is that where applicable and possible, we should also include a statement indicating the volume and nature of the diluent used for the reconstitution, as well as post-reconstitution storage and expiry information based on the stability studies that should have been applied. That's the end of the slide. So thanks very much for your attention, and I look forward to the conversations at the roundtable.
Thank you, Alan, for this presentation, insisting again on all the various points of attention to be considered during the development of a challenge agent, so as to design not only the manufacturing process, but also the quality control strategy, as well as the labeling or the transport and storage condition, and so on. So all these aspects, and as this has already been presented by the previous speakers, these are all dealing with the CMC development aspects. So now I have to take the floor for the penultimate presentation before, but I have to share my screen, sorry for that. So I'm Jean-Luc Trouvin, why I'm sharing the screen, yes. Here it is. So I'm Jean-Luc Trouvin, former chairman of the previous chairman of the BWP at the EMA, and professor at the University of Paris, and especially in charge of the quality assessment for biological medicinal product. But I have to address the question of routine production, which standards or manufacturing, and which standards, if any, to apply. And very frequently, the only term which is used is GMP. And I would like in this brief presentation to clarify what GMP are and more precisely what they are not. Oh, I have to do that. Okay, good. For that, I have first to clarify two important steps in the life cycle of a medicinal product. I have taken the medicinal product as a model for the challenge agent. This challenge agent is like a medicinal product administered to humans with the intention to provoke a disease and study the clinical or biological manifestation and possible effect of pharmacological intervention. So this is why I will take the model of medicinal product exactly as did ILDE, in fact, in their general consideration. And so for this medicinal product, there are two activities in the life cycle to be envisaged. First, the CMC development, and we had already 3 presentations on that. CMC stands for chemistry, manufacturing, and control. And second, the routine phase after approval for routine production and release of batches of the product to be administered. So we have now understood that these are two distinct activities which are included into a wider framework called the pharmaceutical quality systems. And these two activities in the life cycle management of a product are distinct but complementary and contribute to the delivering of a product of quality. What do we mean by product of quality? In fact, a product of quality is a product which is well characterized with a defined purity profile, which is stable while waiting the administration and stable at the time of administration. Of utmost importance, product of quality is a product well calibrated and titrated. This is the key question for a challenge agent, and we have the presentation from Eddie, and with a well-controlled intra and interbatch reproducibility. And all of these quality attributes are essentially to ensuring the reliability and relevance of the trial. How relevant would be a trial if the challenge agent administered is not consistent from one volunteer to another? Now that the model and expectation have been clarified, let's describe briefly these two activities. You first start with your project, with your ID. For example, I would like to challenge a volunteer with a given virus load. From this ID, you can elaborate a profile. This is the target profile or the quality target product profile. And then you start a series of lab experiments, paperwork, in vitro, in vivo, in silico experiments to document the various criteria explained by ILDE and clearly identified by the two former presentation. And at the end, you end up with a declared documentation, an authorized documentation by the regulatory authorities in which you describe the product of interest and its intended purpose, as well as the manufacturing process and the quality control strategy you have developed. All these activities are called the CMC documentation, chemistry, manufacture, and control documentation, and will end up in the module 3 of the CTD, the now international format for a common technical documentation for a medicinal product, which will be reviewed by the competent authorities. But after that, you have to go for the tech transfer and for the deployment of the manufacturing process you have developed. And this will be based on this documentation that a manufacturer will have to implement this manufacturing process for routine production. And in this case, this will be the GMP framework, good manufacturing practice framework, which we'll have to apply. So from this diagram, we can now understand that CMC and GMP are indeed two important parts of the pharmaceutical quality system, but the second part, i.e. the GMP, cannot go without the first part, i.e. the CMC documentation. So I would like to illustrate this issue with a few texts I have extracted from several guidelines or texts which are available, of course. The first guideline I would like to quote is the international guideline ICH Q10, already mentioned by Hilde, dealing on the pharmaceutical quality system. And in the introduction of this guideline, you can see that the GMP do not apply to the development part of the life cycle. And this diagram clearly indicates or illustrates the two activities, development on the one hand and production. And you can see that the gray area, which delineates the activities to be considered under the Gmp rules, does not encompass the pharmaceutical development. Going further in these guidelines, you can see under chapter 311, the role of the pharmaceutical development, which include the design of the product, the design of a manufacturing process to consistently deliver the intended performance. These are other words, I would say, to explain what I have already presented in the previous slides. So now that the CMC development is well understood, let's move to the GMP part of the quality system. As an introduction to GMP, just to remind you, the GMP, i.e. good manufacturing practice, are part of the overall quality system as exemplified in the previous slides. GMP consists in a series of principles and recommendations to contribute to the right execution of a production run, including the quality control test, as already said by Alan, to guarantee traceability of the batch records by implementing a monitoring of each and every production tickets or document or checklist, etc., etc., and to guarantee that the material and equipment, as well as the personnel, are validated or qualified for the intended activities. In other words, GMP, by their set of principles and rules, are by themselves part of the quality assurance system. So based on these principles, let's have a look to some text and guidelines issued by WHO and the EU regulation. For WHO, sorry. Okay, now it's good. For WHO, it's clear that GMP is part of quality assurance, which ensures that products are consistently produced and controlled. I do insist on the fact that in the manufacture activity, the control tests are and should be included and should follow the same requirements in terms of validation, traceability, etc. But also for WHO, the expected quality is as required by the marketing authorization. And so we are back here to the previous concept of CMC first and final document, which the manufacturer will have to adhere to. Among the risk of misproduction, if GMP are not followed, WHO mentioned primarily the cross-contamination or the risk of false labeling. and the GMP WHO guidelines identify the key chapters of a quality assurance system, which will be dedicated to the manufacturing operation and working condition. If we go to the EU GMP guidelines and GMP standards, this is almost the same. First, the definition lays down the objective of GMP, and here again, GMP are part of the quality assurance system. Article 5 of the regulation and the principles insist on the need to follow the process declared in the CMC dossier. Here again, the link between CMC and GMP as to ensure that they are fit for their intended use, comply with the requirements of the marketing authorization. In other words, the manufacturer cannot decide or invent on its own the process to be applied, and more importantly, cannot modify it without prior consultation and approval by both the sponsor of the product and the regulatory authorities. You have here the 9 chapters of part 1 of the EU GMP rules, which enumerates the various rules and requirements in terms of personnel, premises and equipment, documentation, etc., so that every working condition are indeed well specified so as to contribute to a consistent and reliable production and release. Once again, I would like to draw your attention on the fact that this guideline is very close in its principles to the WHO guidelines, that GMP is indeed referring to the organization and framework for the routine production of medicinal product, and not at all with any CMC development aspects. If we now consider all these principles for the production of challenge agents, we have excerpted from 2 WHO documents the viewpoint of WHO on this issue of GMP. First, in the technical reports on human challenge trial for vaccine development regulatory condition, document in 2014 and 2016, one can read the quality of the challenge agent should be comparable to a candidate vaccine, and that regulation for these trials need to be well defined by the national regulatory authorities. So, needless to say that the regulatory expectation will very likely be on CMC and GMP levels. In another more recent document, the regulatory expectation for the challenge trial and COVID-19, the famous blueprint document issued in 2020, WHO is insisting on the need that the selected strains should be sent to a selected GMP manufacturer to have batches of each virus prepared in appropriate formulation for use in a SARS-CoV-2 model. And further, Chapter 11.1.1 addresses the assumption related to the manufacture of GMP batches. So this paper, as well as the 2 others, are coherent with the previous recommendation of adoption of the GMP rules for the production of challenge agent batches. So based on all these considerations and before concluding, what level of GMP should be envisaged? First of all, we have to acknowledge that well-structured and acknowledged systems such as GMP or ISO are good tools to identifying the key elements for production, control, and release of challenge agent batches. In the white paper we are presenting and discussing today, section 5C is proposing the key principles to be followed when setting the manufacturer obligation. We have not to forget that, as recommended by WHO, the level of requirements will be at the discretion of the regulatory authorities. Considering the final use of a challenge agent, there would be no reason not to have a minimal set of requirements, and particularly in terms of personnel, facilities, equipment, analytical tools for the quality control policy, etc. And particularly considering the need for having a consistent and reliable product. This will be discussed with the regulatory authorities, which could use a case-by-case approach to adapt their final requirements, but very likely the GMP principles will be the basis for discussion. So to conclude, it was important to remind that GMP is one of the parameters of the overall quality system for a product of quality. It is also important to stress that the challenge trials have an important added value in the development of therapeutic strategies against pathogen, and as such, a challenge agent should be of quality and reliable. As such, for both parts of the quality system, CMC and GMP, all efforts have to be done using some formalized approach, which have been extremely valuable for the development of medicinal products. GMP rules are certainly a good basis for reflecting upon the key points to be monitored in the production environment using a proportionate approach with the help of decision-making tools such as risk analysis and risk mitigation plan. Thank you for your attention. So I think we are just on time, or no, 5 minutes late, sorry for that. And we can now go for a break. How long, Peter, you authorize for the break?
Yeah, we will take 10 minutes break now.
10 minutes, okay.
We will resume at 25 after the hour. Just one remark for the presenters. We are putting your presentations on our website unless you let us know that you want to change something or you don't want to have it done. Please inform Abby, Charlie of that. So let us take a short break of 10 minutes and we'll resume at 25. Over.
Thank you. So the last presentation, in fact, I was wrong previously. There is a last presentation on the hookworm as a second relevant example, which will be given by Jeff Bettoni from George Washington University, where he's associate professor, and by Linda Shellash from Falling Water, West Virginia, and she is consultant in quality assurance and GLP regulation. Floor is yours.
Thank you. I'm just going to go briefly through the manufacturing process that we use for hookworm, and then I will turn it over to Linda to talk a little bit about some of the quality assurance and quality systems that we put in place. Briefly, we use our hookworm controlled human infection model to test both accelerate the study of hookworm vaccines and also other control tools such as chemotherapies. And as you know, hookworm is endemic in many tropical areas where millions of people are infected. And to have a controlled human infection model for hookworm where you can control the eggs and the infection rate is quite important for our critical path. We're currently supported by the Wellcome Trust, where we're transferring everything I'll talk about today in terms of GMP to a site in Brazil, in Minas Gerais, Brazil, where they'll be taking over this process. And just as I said before, we currently use the hookworm ChIM as a platform to test our hookworm vaccines, but also to begin testing of several other drugs that are for hookworm, such as tribendimidine, Tri-5B, and other drug combinations, and also to evaluate new diagnostics. And hookworm, as you know, because of it's rather an innocuous infection, it's also used as what some people call a living medicine for immunomodulatory, its immunomodulatory effects for several diseases. Right now, I'm going to talk a little bit about our project milestones that we're working on. Everything I want to talk to you today is about our capacity building to move this to Brazil. There's a lot of Brazilians that I see are currently participating in this webinar right now, and I think that's great. I'm going to talk a little bit about the tech transfer, and these are the steps that we're going to do in Brazil for the ChIM. And this also outlines how our manufacturing process, because our manufacturing process actually begins with a human donor. We will establish a human donor program in Brazil. We will then go through a dose escalation study in a Decatur and endemic area to see the level of inoculum that we can infect someone with. And then also, then finally, we'll use the ChIM to test the efficacy of several hookworm vaccines that were under development with us. I think just briefly, I got to tell you a little bit about the life cycle of hookworms, so you'll understand our manufacturing process. As you can see here, hookworms are an infected individual will excrete the hookworms in their feces, where they will then grow out to the filiform larvae, and then they can infect a human again by penetrating through the skin, where they'll be swept up through the vasculature, spend some time in the lung, and end up in the lumen. And hookworm infections can last between 5-10 years. That nobody really knows the duration, but they are long-term infections that take up residence in the human host. That's going to be critical for what I'm about to show you for our manufacturing, actually. So we have a donor program that we've set up at George Washington University. I think what's critical is we have 3 people at George Washington University who are always infected with hookworm. And I think what's critical for them is that they are the subject inclusion and the subject exclusion criteria. It should be exclusion down here, not inclusion. But all of our subjects are all of our donors are between 18 and 45 years old. They're in good health. They have to be available for the duration of the study and willing to consent, but they must not test positive for hepatitis B surface antigen. They must not test positive for HIV infection, and they must not have a confirmatory test for hepatitis C. I should also say that we produce hookworm at George Washington University under an IND, and hence under the auspices of the US FDA. Just briefly to talk to you a little bit about what we're looking for. This is some of the terms I'll be using. This is time to patency, and you can see this Kaplan-Meier graph that infecting someone with 25 and then 50 larvae of hookworm, where you can achieve a 100% infection. And usually the time to inoculum to the first evidence of infection is about 84 days. So when someone is infected with hookworm, you'll see this word a lot in our manufacturing process, which is eggs per gram of feces. We measure it using this saline float. You can also use a polymerase chain reaction. And what we're going to talk a lot about is how much someone is infected. And you can see that initially there's a peak of infection, but then these are chronic established infections, and it goes, the EPG goes down and then remains stable over a number of years. You saw the BSL-3 facility previously for COVID. Well, we don't really have that because actually the donation sample, as I talked earlier about, is human feces that we collect on a weekly basis. This is our BSL-2 plus facility, where we process the feces, and I'll go through that right now, but this is just a room in an entire BSL-2 positive BSL-2 plus facility. This is the actual manufacturing process used for hookworm. We take the human feces, 10 grams of human feces, where we then mix them with an antibiotic wash. They're then put on a Petri dish. The Petri dish is then, we placed a Wattman paper on the Petri dish. We then add the feces onto the Petri dish, onto the Wattman paper on the Petri dish. And what happens is, and you can see an example here, is that the larvae migrate down from the feces into this antibiotic wash culture that's down here. And then we then remove the antibiotic wash culture, and then we wash the larvae several times using water for injection until we clean up the larvae. And this is it. This is done on a weekly basis. So you can see with the part of the human donor is here now, and this is a process that usually takes 4 to 5 days with an incubation period about 9 days. Usually the mean, the eggs per gram of feces for our third stage larvae is, our donors tend to have about 100 eggs per gram of feces on a regular basis. This is the total number of larvae that we expect based on these eggs per gram of feces and our yield. And I'll show you several of these release criteria that we use so that we can use these as an inoculum in a human. So we have three release criteria for hookworm. We have they must be viable or motile, and I'll show you that very briefly. They must be hookworm. There are several other species of hookworm growing up in the lab. There's Ankylostomum salonicum and duodenale. And then the FDA insisted that we test for microbial quality under the USP 61 and 62, and each lot that we make of hookworm has to be tested for this microbial burden. So this is our motility test, kind of like our version of a potency test, and it's just to look to see under the 40x magnification. You would just count the number of worms in the field, and then at least 90% of them must be viable, and viable, as you can see here, is simply a very simple the movement of the worms. Each lot is then we remove several of the larvae, and we put them through the speciation test. What we did for the speciation test is we just created a very simple genomic test. We synthesized this Nicator americanus DNA, but we added several base pairs, so we know it would be the synthetic version. So this is your positive control, but we know it's the positive control because it's going to come in at that base pair weight. Since we have Ankylostomum salonicum in the lab, we also did an Ankylostomum salonicum DNA, which you can see right here. We have a negative control, and then we have the actual test sample, and this is where the test sample should appear. So this is our equivalent to a vaccine release identity test. So each lot has to have be passed with this genomic test. The final test, it has to go out of the lab, and we send it to a vendor. And the vendor then usually takes about 2 weeks to test for Staphylococcus aureus, Pseudomonas aeruginosa, and they will then send us back a form that says the larvae did not have any microbial burden, and then we can proceed to using it as an inoculum. So we have developed for this is our release form that we have here, our certificate of analysis. You can see that before we inoculate anybody with hookworm, the third stage Americanus has to have its identity done by the polymerase chain reaction. The hookworm have to be viable at harvest, as I showed you before, and the microbial quality must be negative as identified by the vendor. This accompanies every lot of hookworm that's used as an inoculum in our challenge studies. This is just briefly to go over. This is, we almost make lots once or twice a week. I had to take, this is a very small graph of a much larger process that we do. So you can see on a weekly basis, we're doing the testing. Does it pass viability? Does it pass the microbial burden test? If so, then it is used to inoculate a human. This is just simply the preparation of the inoculum here. I showed you the worms previously. They're then removed. They're put on a gauze pad. They're placed on the arm of a volunteer or a participant in the trial. And then you can see the skin 1 hour post-application. And this is just another, so what usually happens at 17 to 14 days post-application. So I'm going to let Linda now talk about our quality assurance testing in GW. And so I'll stop sharing, and Linda, you can start sharing.
Okay, thanks, Jeff. Are you able to hear me okay?
Yeah.
That's okay.
All right. I am a little bit new to all of this, so I trying to get my.
Would you like me to share the slides for you?
Yes, that would be wonderful. Thank you.
No problem.
Okay, and we can get out of the and put it into the slide. Yeah, there we go. Okay, well, thank you for this opportunity to talk about how we implemented our quality systems for the CGMP compliant production of Necator americanus third stage larvae at George Washington University. Back in 2017, when we first began our journey into this project, we knew that we needed to implement a quality program that would meet our US FDA requirements. And we took a look at these requirements. The first one being the statutory requirement under the Federal Food, Drug, and Cosmetic Act that anything administered to humans would meet certain efficacy, quality, and safety standards. And so that is our overriding statutory requirement. We looked at our CGMP regulations, which are of course for the full-scale manufacturing of drugs and finished pharmaceuticals, and also a relevant guidance document, which became the primary basis for our George Washington University program, and that's the Guidance for Industry for CGMP for Phase 1 investigational drugs, which was the stage at where we're at. So that was our primary focus for implementing our program, and it provides for certain leniencies from the full CGMP regulations based on the level of development of the product. So our objectives were to assure that we had quality built in to our process through the proper design, monitoring, and control of our manufacturing in order to produce a product that was consistent according to the quality standards that were appropriate for its intended use under our IND, that the product would be safe for human subjects, and that the identity, strength, quality, and purity characteristics of the product would be assured through the adequate control of our manufacturing operations. So in order to achieve our objectives, we looked at the critical areas of compliance for our Phase 1 product. We identified the need for robust, approved standard operating procedures that would address both our general quality system SOPs as well as our production-specific SOPs, the SOPs that we have in place for the actual production process, the testing, and our product release procedures. Validation of critical processes and our test methods to make sure that they were appropriate for what we were intending to do. Our sample and raw materials management processes, our records and documentation in order to assure the appropriate transparency and traceability of our whole production process. Environmental monitoring is relevant to our product quality within our production facilities. The adequacy of our facilities in order to be appropriate for the production, the size, making sure that certain activities were segregated when there was any potential issues with cross-contamination, the appropriate environmental controls, primarily temperature, as well as our cleaning procedures, looking at our equipment in terms of capacity, functionality, maintenance and calibration requirements, and also for situations where we would need to have dedicated equipment in order to assure product quality. How we were going to have deal with certain things that would ultimately happen, out of specification results potentially, in any deviations and unexpected occurrences, making sure that we were assuring our personnel qualifications through our training program, and also defining what the quality assurance oversight would be. One of the first steps in achieving our objectives was implementing a quality manual in order to define the overarching policies and procedures within our facility that would assure that quality processes are in place, addressing our organizational structure, our facilities requirements, how we manage controlled documents, just all of the umbrella framework that you need to have in place. Then we began implementing our general quality systems SOPs to address subjects such as the SOP approval and management processes, our training programs, our documentation requirements, handling deviations and having a corrective action and preventive action plans, equipment use, calibration and maintenance, QA monitoring, as well as our retention and record and management of our records. At the same time, we were implementing our production-specific SOPs that included the raw material management. Everything that comes in for use in the production process goes through a quarantine review process where we examine the C of A's and make sure that they are what we need in order to be a raw material in the production process. Our reagent and solution preparation and labeling, we have specific solutions, the antibiotic wash culture and antibiotic wash harvest solutions that we have to prepare for every production batch that we are preparing. The receipt, handling, and storage of the fecal specimens that come in through our donor, our sample management and labeling requirements, the primary larvae production process, where the process that Jeff was showing in his slides, our testing procedures, the larvae viability, our PCR for the species identification, as well as our microbiological quality, which is done by our subcontractor in accordance with the USP requirements. We also established SOPs for our lot release procedures, which included the approved specifications, the pass-fail criteria for releasing a lot of our products. And important to note that all of the SOPs include approved worksheets and forms in order to assure that we have the necessary documentation that will remain after our production process is complete. Just a few words about our lot release process. Every lot that is going to be used in our clinical trial undergoes a formal release process. All of the production records, the testing results, as well as all supporting records and the certificate of analysis are audited by QA. The supporting records include our raw materials documentation, our equipment records, our training records, all of those general quality systems things that pertain to that particular lot. A formal audit report is issued. All findings and deviations are addressed and closed out before the release process can be completed. The certificate of analysis is issued. There's a 4-level review process involved with closing out the C of A, the preparer, the reviewer, management, as well as QA. And the final approval and product release is completed by QA. And just as a final point, just stressing the importance of our good documentation practices. They are really critical to our whole process. This is a controlled and verified process to assure that the product is consistently produced, to assure its purported ISQP characteristics. And this documentation provides the only enduring evidence that we adhered to our procedures and that we adhered to the regulatory requirements. It allows for reconstruction of our manufacturing process and demonstrates that required quality processes are in place and were followed during our lot release program. That concludes my high-level discussion of our quality systems, and I appreciate this opportunity to present. Thank you.
Thank you both for this presentation, which is indeed a second example where the medicinal product model is used, acknowledging, of course, the need for some adaptation. So this concludes this session with a presentation on practical cases and also the general consideration which are laid down now in the white paper. And maybe it's time now to open the roundtable discussion. So
Yeah.
I give the floor to you, Peter.
Yeah, I will give the floor immediately to Isabelle Beckeret-Janding from Polelic Institute. But before doing so, all people attending this email could use the question and answer box to put questions to our panel. Who do we have in our panel? We have a number of CMC experts. We have Wim van Molle from Cienzano, who is the OMCL in Belgium. We have Paula Salmikangas from NDA, who is an ex-CMC regulator. We have Dean Smith from Health Canada. Yetsumon Prachumsri from India. I hope it is not too far from the right pronunciation. We have Pauline May from the University of Leiden. Erin Kari Boanteng from Ghana. And we have Scott Stibitz from the US FDA. We have Winfred Badanga from Uganda. And then the other speakers, of course, like Jeffrey and Eddie, Hilde, are still with us. If there are questions, they can answer them. I forgot, sorry, Peter, I forgot you to mention. You are there as well. Peter is from the MPA, the Swedish agency. Okay, I give the floor to Isabelle.
Yes, hello everybody. Welcome to this panel discussion. I think we've heard a lot of really interesting talks that filled all kinds of guidelines with life and practical conclusions and considerations. I think that's the most important thing about these meetings. And I think we should just head off and maybe discuss a little bit about the manufacturing. There's a lot of requirements. I think it was you, Jean-Luc, who said that basically you cannot choose how to manufacture. So is that really true for a human challenge agent? So maybe I could pass on to Hilde.
I'm sorry, I was on mute. Yeah. So basically for the manufacturing of a human challenge agent, of course, as the types of human challenge agents can be quite different, not everything is applicable to all types of human challenge agents, as what we said before. But I think there are some general principles that needs to be followed. And if we look at the white paper, everything that we have described under the section 5A and 5C with regard to the selection, characterization, and proposed testing, like testing for advantageous agents, etc., are things that we have to do, whatever is the human challenge agent that we are developing. And I think this was also illustrated by the examples, because both for the SARS-CoV-2 example and for the hookworm example, I see that this is in line with what the producers of these challenge agents did. So this would really be my recommendation also for the future and for the development of new challenge agents.
Thank you. Would anybody like to further comment on this? Is there any regulatory viewpoint on this?
Maybe I can just add, waiting for the reaction from regulatory people. I could add that I think, again, the model which has been presented and the formalization of all these checklists, I would say, of point to be considered in the development as well as in the implementation of the routine manufacturing process should be used as a basis. For sure, we will have and we have to consider some adaptation, and we had with the hookworm, for example, some adaptation needed. But I think that we should have in mind the principles, and after that discussed on the principles on how and to what extent we can adapt these principles to the specific situation.
Very true. And I think the most important thing is always to keep in mind, what are we going to use this for? We're going to use it for creating evidence in a clinical trial, and we need this to be robust, reproducible. So may I again ask one of the regulators? I don't know, Dean, Jetsamon, Eric, would you like to say something about this and how
Hi,
we
I'm not the regulator, and but
Sorry.
normally I'm one of the researchers that also very glad to see the white paper, because that is a guideline. But the challenge to the researcher that have to use or to produce the challenge agent, because I work on malaria, Plasmodium vivax, that you cannot culture. So there are a lot of things to consider, but the white page help, outline the category that I have to consider step by step. But I think it's challenged to think about in the detail, specifically for my my reagent that I'm going to produce. But it's very good.
So would you think you need more detail, or do you think with this as the base you can work it out?
I think the basic is good enough, but it's specific thing. For example, how to test the quality of your batch. It may not have that simple thing, because malaria is specific to human. If you want to check the challenge efficacy or the efficacy of the parasite after you produce many lots, normally in general, the research now is not up to date that we can culture parasite in vitro to check the viability, for example, like that. So that's the challenge with to think about the quality from batch to batch, how you're going to do, because you don't want to every time inoculate to 1 or 2 volunteers to prove that the dose that you use from previous study from this lot will give the same parasitemia in your volunteer or something like that. So I think that's the challenge. But the guideline that you put is very useful, that make us think even more in terms of the technique they can apply for our reagent.
Yeah, if I may
Doris? Sorry, yeah.
Okay. Yeah, I mean, I read through the document. The guideline is a good document, but then there were one of few things that I noticed that were, missing, and I could also see that from the presentations, especially if it's a virus and you are growing it on a cell line. And I didn't hear anybody talk about the end of production cells, because the time that you have is, you should be able to show that your cells have maintained their integrity from the beginning to the end. The integrity of the end of production cells have to be checked, because if the integrity is not maintained, mind you, host cell protein profile can change the quality of your drug substance. So that has to be checked. And I didn't hear anything about comparability. I mean, there was no talk about a scaling, because if you scale up, that the size, the difference between the batch that you use for your clinical challenge and your commercial, if they are different, you would have to do comparability. in accordance with ICH Q5E. That one is one. And you go through the document again. Some said manufacturing non-GMP non-GMP environment. As a regulator, I don't know the definition for what is a non-GMP environment. Okay, over.
Thank you. Would somebody like to comment on this? Wim?
Hello. Oh, I'm sorry.
Yeah, Scott, go ahead. Go first.
Okay. I have background noise for which I apologize, so I've been muted to reduce that. I just wanted to say I like what Jean-Hughes said, that I think the important part is to stress the principles because the area of challenge agent manufacture is so varied, and each one is so unique. I don't think it's going to be possible to have sort of a prescriptive approach to this. Not to say that the paper can't provide examples and frameworks, but I felt today, listening to these talks much more than the 2019, that we're much more in harmony. I think we're using completely different language, but following the thread, I think we're in the same place. That GMP for challenge agents is not necessarily full scale GMP, but again, it can depend on the challenge agent itself. It seems like in the area of a COVID challenge agent, maybe that's much more important. But just again, to stress the principles, which is really the message in our phase 1 GMP guidance document. So that I'll stop there.
Thank you. Wim?
Yeah, one point we should not forget is what Isabel, you said first, that the intention of this this agent is to test in a first in human. And then when we are, let's say, all talking about the manufacturing, we should always, or we should not forget the scale we are looking at, because it's for use in a clinical trial. So it's not immediately the intention to produce hundreds of millions of doses, as we have now the case for the COVID vaccines, for example. And that what we have seen from the two examples, the hookworm and the COVID, I think those are, well, I could consider them good models, because there we have indeed seen that all the testing, especially also for the adventitious agents, has been well performed. And of course, if you're looking, or if manufacturers or developers are coming with different agents or different types of organisms even, then this should always be looked at as a case by case, because as said, we are focusing on a first in human, and their most principles is the safety of your agent, either whether it's the candidate vaccine, the IMPD, or it's challenge agent. And we know also that at least for first in humans, the requirements on GMP, or the requirements in general on quality, are far from what is required from if you would go for a marketing authorization. So it's always to bear in mind that it's not the intention to license the challenge agent, but it's to license the vaccine which is used in the challenge study. Thank you.
Joris, I think you have been watching the chat and the Q&A. Is there any questions we should pick up here?
Yes, indeed, there is a question of Professor Robert Zauerwein relating to what Wim just said. I read the question: regulatory approval will be required for batch release. Experts are qualified according to categories for medical devices, therapeutics, medical diagnostics, etc., etc. These all have different criteria. How do we present a challenge agent in the regulatory environment? Between brackets, partial stumble block within the Dutch system. So, Wim, you surely have an idea about that, and I address this very important question to the panel.
Well, I can start with this maybe. Well, it's a very good point, because as we know, this is not a medicinal product. So now about the problem in the Netherlands, where you have, let's say, more categorization of the products or the different agents under assessment. Well, for Belgium, I can say that people involved in clinical trials are also people involved in assessment of dossiers for marketing authorization. Of course, it's true that how should we look from a regulatory point to, as I said before, to a challenge agent, which is not a medicinal product, but what we have seen from the example from Hookworm, at least those manufacturers are characterizing and are testing their challenge agent as if it would be a medicinal product. So they are putting the barrier at the same level. So I'm not sure whether this is really answering the question, but I think we should, in the best possibilities, assess this as closest as it would be to a medicinal product, but always bearing in mind this is in clinical trials. It's not the same requirements as would be needed for a marketing authorization of a medicinal product. But maybe some other people can have different looks from across the ocean. Thank you.
Could I ask you, Dean, maybe you have a little bit more of a global overview? How would you see this?
So thank you, and thank you for the invitation to be here. So I agree with all the comments made so far. Is that and in Canada, what we do, again, we don't have a great deal of experience with challenge reagents as such, but we have had a lot of discussion about this internally and watching work that's gone on elsewhere, both in Germany and the US and others. So if it were to come into us, it would be put into the most relevant group to deal with it, and we would recruit the expertise required to deal with the specifics of any particular pathogen. That's really the only way that could be done. So and I think this is a challenge for regulators as well as a challenge for manufacturers, these reagents, is to be very open-minded. I think all the comments made about the document in terms of its structure around principles is really very positive, rather than trying to be too detailed, too prescriptive. That is a very dangerous territory. I think the examples chosen today have been very good because they cover everything from something that is closer to a routine manufacturing process, say for a virus with the COVID example, to hookworm and others, which and considerations around malaria agents, for example, which are sort of out of the typical range of things, but certainly can be managed, providing a regulator is sufficiently open-minded about the principles and not the specifics of any one regulation. So for clinical trials in Canada, we require materials to be comparable to the stage of development, so phase 1, phase 2, et cetera, where we don't require full validation of all of the processes, but we certainly would ensure that the assays are fit for purpose, and where possible be fully qualified, or actually be fully qualified at least, but perhaps not fully validated in the routine sense of that, but certainly fit for purpose. And all of the conditions around GMP, maintenance, SOPs, document control as mentioned by the examples, all of those elements really must be in place to ensure integrity of material, but it has to be realistic to the stage of development. That's the key piece. And just a comment also on Eric's piece about comparability and scale up, but again, in light of what Wim was saying, we're not going into large scale manufacturing, but to the extent you scale up, comparability would be essential. Over.
I would just, this is Scott, so I would just second everything that's been said. I think we're all on the same page. GMP for challenge agent is not, the intention of that is not the same as GMP for a licensed product. Over.
Okay, we have a remark from Paul Kay from MHRA UK. Of course, they do not regulate challenge agents per se. I don't know whether someone can react to that. And in the same vein, 2 remarks from Martin Schutton. For example, do regulatory authorities have difficulty in judging aspects when challenge agents are concerned, compared to attenuated or non-live vaccines, for example? So I understand from this question where he uses the example of live Shigella, where is just the border for a challenge agent compared to a live vaccine, for example. And he also asked to look at that into the white paper. So I launch it to the panel. I don't know who, Isabel, will react to that.
Would you feel that this is your area?
Talking to me, right? Well, I think I'm a bit repeating myself that it all depends, as also Dean said and Scott said, on the stage of your development. That whether you're going for a first in human or whether you're going to maybe phase 2 or phase 3. Now that's about comparing your challenge agent to what you would require for example, an attenuated strain. Yeah, it's difficult to say, but I think this is again depends on what you would require, because if you're going with the first in human, what you will require for your attenuated will also, let's say, grow in the further phases. And again, for the first in human, the effort is always put on the first, let's say, action is always put on the safety of your product. So if your challenge agent, or rather said, if your attenuated vaccine strain should, let's say, answer to a limited or a minimum rather set of safety requirements, then also your challenge strain should be responding to this. Because there is also, you could also argue, why would you not make an attenuated virus itself and use that as a challenge strain, maybe to work on or to look for other or better candidates. So let's say, to me, I think those two can be put at the same level of requirements, but always bearing in mind again that you are looking at the clinical trial and not at the requirements of a marketing notation. We have seen this also now more actually in the COVID vaccines, that also what was required for vaccine candidates in the trials was maybe even less than what was required from a normal situation.
Peter.
Of course, except from the let's say the safety requirements. Thank you.
Peter, maybe you could give us an insight whether you would see this similarly and whether you see this relationship, or is this two different entities?
Yeah, we haven't had any clinical trials involving challenge agents in Sweden, to my knowledge, but so I'm not actually quite sure what requirements we would have on that, but in my opinion, it should be the same requirement as for an investigational medicinal product that's used in clinical trial, including GMP. So I think that's extremely important for this type of agent where safety is extremely important, of course. So that's my view. And I think after listening to those the presentations and including Jean Hugues, I think everybody seems to agree about having the same quality requirement as for a medicinal product, actually. Although it could be certainly questioned whether this is a medicinal product. I wouldn't think so, but that doesn't matter, actually. We should have the same requirements.
Anybody else who would? Yes, yeah. Scott?
Yeah, if I may come in, and looking at the scenarios, yes, if the challenge agent is linked to a vaccine and it's in a clinical study and it's in phase 1, yes, then I do agree with what Wim's saying, but then it should be manufactured under GMP, but then what type of GMP? Because phase 1, yes, the product should have clear release specifications, the manufacturing process should be well defined, but of course, as a regulator, if it's phase 1, I'll not go and asking for process validation or analytical method validation, so that's where the difference is. But then the basic GMP requirements would have to be met, at least the starting material should be well defined, the manufacturing process well defined, and then the challenge agent itself should have a release specification which can assure the quality and safety of the product. Over.
I think it highlights the importance of these trials in a way, because if you ask for these very high requirements, you need to think long term, and you need to be planning long term. It's not going to be a quick shot.
Yeah, I just a comment. I think I heard you talked about the process validation, and that's not a requirement, obviously, for investigational medicinal product in the clinical trial. So that's not included.
Paola.
Yeah, if I may say, I think the same. I fully concur what Jean-Hugues was saying, and what has been discussed here about the similar requirements as for medicinal products. But even for medicinal products, the requirements are not the same for each and every product. They vary depending on the risks. And here I would say that the risks of the challenge agent should dictate how much information there should be. And of course, when we think about the GMP requirements, one very important risk in my opinion is the release of the infectious challenge agent to the environment. And for medicinal products, there is an environmental risk assessment necessary for every medicinal product. So I would say that the requirements for GMP should also take into consideration the environmental risk aspect. But overall, I fully agree with Peter that the requirements are more closer to the investigational medicinal products than for the MAA BLA. Thank you.
Scott, I think you wanted to add something.
Yeah, I was just going to speak to the issue of medicinal product. As I understand, using the translational guide, that would be what we would call a drug. And so challenge agents are considered to be drugs by the FDA, not so much because they're intended to, diagnose, treat, prevent disease, but because they affect the structure function of the body. So again, it sounds like we're really talking about the same things. We're just using different terminology. And just to address something that Paula said, I mean, one of the ways we typically deal with environmental issues, if a challenge is done outpatient, you have little control. But for inpatient studies, it would be typical, for example, in a diarrheal disease challenge, that patients are not released until they've had 2 negative stool cultures, and with or without antibiotics. So, I mean, there are ways that you can approach that. I don't know if that is something that you would want to include in the white paper. That's all.
This was also a question from the audience. So is there another comment maybe on transportation, storage issues, and also the waste disposal and destruction?
And if I may, there is also a question from Wan Lapa Roopsong on stability, and Martin Schutten in the same vein on quality parameters for release.
So who?
Can I pick up the stability piece? I did see that in the chat. It was asking about frequency of testing. So at a core, a key element of all this, and it goes back to many other comments made already, is characterization of the material you're making is really rather central. And that, as Jean-Hugues covered in his piece, there's the chemistry manufacturing development phase, and the knowledge that the manufacturer is going to have, or the investigator is going to have about the material. And so therefore, an understanding of the material and its characteristics will be central to designing a stability program. If you're dealing with a very stable material, or you're going to keep it under conditions prior to use and very short intervals in use, and you're keeping it stable at -80, whatever, then your stability testing is you'll need something more focused in use conditions if it's very fragile, and of course sufficient ICH type of guidance around general stability assessment. And there was an example of forced degradation studies And since Eric mentioned comparability as you're going along, something that we're very focused on at Health Canada within this context, and of course in manufacturing in general, is the use of forced degradation to assess things, including through scale up. You can get very quick insights into changes in stability through manufacturing and scale up using forced degradation, much more so than you always have to do the real time as well, of course, but you can get insights very quickly to changes in stability. So those are some key pieces, but the specific timing of your stability testing should be a function of the product you're working with. I mentioned ICH, but rather than simply looking at ICH, that's a framework. You should be designing your stability studies around the properties of the material you're working with. And if I could, just before we move on, I want to go back to a question raised earlier about, say, malaria materials and not having good in vitro means of looking at certain characteristics of that material. We fully understand that, and that's something a manufacturer or an investigative group should make very clear to their regulator, is what the limitations are in terms of the system they're working with. But again, I'll put the challenge back to the manufacturers or the investigative group, is to say, what are the indirect measures that you could establish that would correlate with what a human infection model would show you, ATP levels, other indirect or direct measures of viability, because the quicker you can get to an in vitro definition of that in a robust way, the better the control you're going to have within your system. A human, of course, is an animal model by definition. Animal models are quite variable, so the quicker you can move away from that, fine. If you're limited in terms of timing of development of your process, then I don't think a competent regulator will prevent you from doing what you must do. But it's always in the interest of a developer to find those non-animal model-based potency viability assessments as quickly as possible. That will help your development along the way, and that's a scientific objective as well as an ethical one as well. Over.
Thank you. Alex, I think you wanted to say something?
No, just that I can concur, Dean, in terms of that element can apply to other organisms other than malaria as well. Norovirus is not exactly the easiest to confirm its potency and viability, except essentially in a human infectious dose to characterize that. So in other words, over time it may be that if you're not doing regular infections and you're not knowing that your batch is consistently giving you the same result, if you've got a 3 or 4 or 5 year gap between your studies, you may need to do a small characterization study in a small number of subjects just to make sure it still has its HID50 rather than its TCID50 or its other platform that you use to quantify and potency.
Pauline, maybe could I give you the floor to discuss a little bit about COVID-19 and how this relates to it?
What exactly do you mean by that?
And these issues that have been discussed, I mean, we somehow lost track of COVID-19, so I thought maybe you would like to bring it into the loop and discuss a little bit of what the special challenges are in views of these issues that we've been discussing here.
Yeah, of course, a lot of issues have been discussed the last few minutes. So, yeah, so what we've done, like I already said, so we start the whole process like we were manufacturing medicinal product. So to be sure, we could do everything on the consistency, on the safety, the quality of our production process. So we could do what was possible. And in that way, we started this whole process and we decided what to do and what not to do. And of course, we didn't do it alone. So we sought for expertise from abroad. So in this case, people which were had expertise in the production of viruses, as medicinal products, etc., etc. And with all of that, we have made our decision on how we would do the manufacture, how we do our release testing, how we did the transfer from the research to GMP, etc. Yeah. So, but it's difficult. It's difficult because also the challenge with regulations, that's also a difficult question. So that altogether made us to make this decision, and especially because for this, the timelines were very strict. So it was something what has to be done as soon as possible. So for us, it was at that moment the best way to do it. And I think, yeah, it's a good way to go forward with the challenge agents like we've did, yeah. And this made, of course, this was an agent which we could produce in our facility, in our GMP facility. So it really was easy to use the whole quality system around it, because when you have an agent, for example, like Hookworm, which cannot be produced in the GMP facility, the clean room which you have available, it's of course already a different story how you're assessing the whole process, at least in our experience.
So I think what is really important is to understand that you do need some time, you need some experience with the challenge agent, and you need to learn how to work with these pathogens. So this is important if we think of pandemic preparedness. Next, pathogen X, this will always take a little bit of time, if I understand you well.
Yeah, and I think what you say is very important. So it was really important in this whole case, though, that the virologists, which were now in the institute, which had a lot of experience with the coronavirus, also with this virus, and they really were very important in the whole team to set up this whole process.
Thank you.
Can I ask you something, Isabel? I have a question for Winfred. Winfred has been also attending this meeting from Uganda, and what she is thinking about this kind of COVID-19 development. Over.
Yes, Peter, thank you for that question. So regarding the COVID-19 development, I should note that in Uganda, we're just having the first ever challenge study. It's yet to be implemented. It's on schistosomiasis. So having a COVID-19 challenge study, I know it would be, challenging, and so many questions would be raised by not only the stakeholders and the regulators, but even the general public. So it's amazing to hear that countries are setting up such challenge studies in the UK. But regarding the document, I should mention that this is generally a good document. It's going to provide guidance to not only the regulators, but also to the national agencies that regulate research. For example, in Uganda, challenge studies are not regulated by the drug authorities. So these are not taken as drugs. So it falls back to another regulatory body, another national agency that oversees all research. So this guidance document will be handy and will be helpful and will provide guidance to the national agency. Maybe what I can also recommend to make it more inclusive and adaptable to all the agencies, can we not only refer to national regulatory agencies or drug agencies, but also include applicable national bodies or agencies that oversee research in their respective countries. Thank you. Over.
Very interesting. Thank you.
Isabel, could I just jump in back on
Sure.
the COVID topic again, and I'd be interested in Pauline's reaction to some of this, is that while the idea of human challenge studies with COVID-19 or SARS-CoV-2 virus has been difficult for many authorities to contemplate for all the reasons we know fairly well, we're, with the advancement of the UK study, and the establishment of that model with that construct, although we don't know the details of the construct, and that's fine for now, is that we're moving into a phase with the variants where very openly, leading agencies are talking about the use of human challenge studies in comparator models from, protection established already through clinical trials with various vaccines, with the original Wuhan strain or things like it. To some of the more challenging variants now evolving. And there was a question in the chat about variants as well and how we develop new materials. So I think we need to be readjusting our reticence to think about SARS-CoV-2 challenge models because they are identified by many groups now as a very potent tool in this new variant assessment scheme. And the ability for manufacturers to pivot quickly and make additional challenge virus and be encouraged to do so is very important. That's a funding issue that was raised in the chat that I can't as a regulator comment to, but I think, and there is a lot of nervousness in many agencies around doing them, but this is now a different window, and the experience we can generate using the UK kickoff and others, hopefully, including Pauline's group, will be very, very valuable. And one very technical question, Pauline, is that through the WHO animal models subgroup, it's been very clear that, you know, cell line selection for your production of whether it's a animal study or for a human challenge study is very critical, because mutations do develop during production in these strains, and selection of cell substrates is rather critical, as is what you showed, the confirmation by NGS that you have in the end what you started with or what you want as your strain. Any comments from you about your process to select the cells and the verification process?
So like I said it in my presentation, we really looked at the mutations which were introduced during the whole culture of the viruses. So we really selected for a strain and a cell line, and also the culture medium, which is also a variant which seems to be which can induce more mutations during the whole culture process. So with that all together, and that's really something which the virologists, which we have a lot of experience with all the culture of the coronavirus and also with the SARS-CoV-2 virus. So a decision which we made together with them, and they really made the advice: this is a strain which you should use, this is a cell line which you should use. Then, of course, we had a cell line which was not GMP grade, so we had to look for a GMP grade cell. So we had to change for a cell line again and again. We had to make that assessment: is the mutation rate not too high when we're culturing this virus in this cell line? And in the end, we ended up fine, luckily enough. Yeah. But really is something which should really assess very carefully. Yes.
So if I may, does this address the question of Adrian Waldfire, who just touches the variants and the variation of strains? And he poses, as Dean already said, the thorny issue: who would pay for updating the agents every time when it's possible? And there we come to another question: when is it time to adapt to a variant and when not? It's not an easy one. Probably a lot of this has already been answered. I don't know whether Pauline or something else, someone else has something to add to that. Otherwise, we can go to a question about GMP.
Maybe one comment. I think the question is, of course, can you speed up this process of manufacturing a new strain once you have experience with the agent itself? And I think this is an excellent example to think about the variants, because that's exactly where we now have to think about human challenge trials, because we're looking out for possibilities on how to license vaccines against the variants, and we're discussing all kinds of possibilities, and this is one. So the question really is, if we see the dynamics of virus mutation, can the manufacturing of the strains, keep up with these dynamics, or is this something more stable and more long-term?
May I pose a question? How far can in vitro, as has been mentioned already, keep up with this issue?
Maybe I'll respond to that, Joris. So the in vitro studies in terms of neutralization studies and binding studies and such that are going on with variants are very insightful. And Isabel's referring to the immunobridging studies that could potentially evolve out of that. I think I'll make just a quick comment. Of course, WHO has indicated pretty clearly, and with agreement from agencies like EMA, FDA, and certainly Health Canada is aligned with that, thinking that at this point, at least some of the more potent vaccines we have out there are, and perhaps all of the vaccines we out there will still offer good protection against hospitalization and serious disease. So there isn't a need to change now, but there is absolutely a need to be exploring alternatives. And what is now widely understood is that constructs modeled on the B1351 mutant seem to be, I should stress, broadly cross-protective, at least in terms of those in vitro studies and some of the initial animal studies as well. And that would be a very valuable exercise if we had constructs made to evaluate that in, for example, a CHIM as a small-scale, quick study as a possibility. That means that we have to fairly quickly adapt groups like PODENs to manufacture an other strain potentially as a challenge material. And that's something with it, again, the funding is clearly off the table of regulators, but is something that if regulators are more willing to consider those types of studies, that would probably assist those funders in dealing with the groups capable of making those materials. So that's just a challenge I put out to regulators, including Health Canada as well, of course. And I'm not speaking for the agency, I'm speaking for myself here.
Thank you.
And
Yes.
and just to bring it back to the actual the process of manufacturing a challenge agent and how that applies to it, I think once you have got an initial process, you've done all the groundwork, you've got that all ready for the for an earlier isolate. If you've got all the documentations and all the principles in place that we've discussed today and in the document, it can make it much easier to reproduce that. You may have to do some tweaking here and there, and of course there are some complications, but it can make you shorten your time frame at least a little bit in terms of manufacturing. So I think it exemplifies the need for some of those principles to be applied, even if you're doing it in a non-GMP facility, if you're doing it in a GLP facility or in an academic laboratory. I'm not specifically talking about SARS here, I'm talking about generally if you're trying to do another run with a new variant or a new batch lot, having that consistency and having the process in place is obviously beneficial for shortening timeframes as well.
So we have, sorry.
That's also a very good example where we could take benefit of the development work which has been done on a given strain, and after that to apply this development work and the experience gained from this development work for a new variant of the strain.
If I could just add to that. So Alex, what you've described is a platform manufacturing technology that you've already honed, and as Jean just referred to, you have many of the validated steps in place, and to the extent there might be some different growth characteristics of a new variant challenge material, you have to accommodate that and obviously adjust your assays to deal specifically with that variant, et cetera, to characterize that. But yes, the regulators have been extremely good in general about using platform technology knowledge, as Jean was referring to expedite decisions. And this has been a huge, it's always been the case to some extent, but it's now sort of more universally so. And that would certainly be applicable to evaluations of challenged materials as well.
If I may, there are two questions left on GMP, one of Jean-Marie Preot from IABS EU. He poses the question whether a GMP mock inspection could help and advance the GMP requirements or GMP-like requirements. And then we have again Paul K, who poses again the question of the GMP need, and in his point 2 in their discussions with CMOs, they are very averse of being asked to make some GMP-like. So I think we still have 8 minutes. May I launch those questions to the panel, please? Isabel?
So who would like to volunteer to answer this question? Inspection is an interesting point. I think it also requires expertise. So I'm not sure a mock inspection makes sense, but let's discuss. Anybody have any feelings there about it?
Now, may I ask, what's the definition of a mock inspection? That means you inspect, point out the deficiencies, but they will not issue out a report then for them to correct it before you come again, or what? Is that what it means by a mock inspection?
I think, as it is written, a mock inspection is something you do. For example, you see an institute that will produce a challenge agent, and you in advance go already to assure that everything is all right.
So let's ask the manufacturers. Alex, any opinion on this?
I would say 100%. You check who you're going to subcontract to do your manufacturing. You want to know that they're going to do a good job. And I think a lot of it also is that, just from experience with viruses, is that a lot of the manufacturers aren't used to using, say, viruses and using an infectious agent. And so you need to work with them to work out how you implement your plan within the GMP facility. But you, of course, want to make sure that their processes, if you are using a GMP facility, then that their processes are adherent to the GMP criteria, and you want to be reassured that before you go into the actual manufacturing process.
Jetsumon, would you also see that would make sense in your case?
No, because for my reagent, it is very far from the GMP. So we like the idea of practice, follow the guideline, but it's impossible to be GMP for now until we can do the in vitro culture of the parasites. So far, still not that close. So I don't think for my case it would be helpful.
Sorry, just to clarify as well, I was saying when you are using a GMP. So in that specific situation, when you're down that pathway, whoever you're using, even if you're using an organization that's not necessarily following a GMP, it's just being reassured that they are going to do, they have what they're claiming that they can do, they are doing, and making sure, because at the end of the day, you're still responsible for your agent. But no, I completely agree with you, Jessamine. Sorry, in terms of the where it's not a GMP manufacturing, you clearly wouldn't do a GMP inspection. No.
Isabel, can I just jump in there? And so one thing that's very clear in our guidance and most agencies' guidance is that it goes to the idea of mock, but I take Eric's point very well, is that what you do want to have is early engagement with the regulatory agency you're going to be dealing with, to explain your situation, explain the limitations of what you're trying to achieve, and as Alex is referring to, the conditions under which you'd be manufacturing, who would be doing that and what would it look like. So early engagement with your regulator is a very good idea. Whether that would entail an inspection is another whole area, to Eric's point, but some clear engagement is very helpful. And I'm sure the FDA will be open to that, as well as other EMA and others, is that it's early engagement is always a good idea.
Yes.
Peter, you raised your hand?
Yep.
Yeah, just from a regulatory point, I agree with, I think Alex mentioned, I mean, if you are going to produce it according to GMP, the facilities obviously must be either inspected by regulatory authority or having an audit, or you can call it a mock inspection, an audit from the qualified person responsible for the release of the products used in the clinical trial. So it's some sort of audit or inspection is mandatory to call it GMP.
Yes, but then I have a question. When you say that most of the authority do not like the word GMP like, but if it's not GMP, then we call it non-GMP, not GMP like.
No, I totally agree. I don't like the word GMP like either, because you could of course argue that sometimes you say that to have a facility according to GMP, it must be inspected by the regulatory authority. I mean, that's not, you could of course think of, you can have GMP and it's inspected by a qualified person also. So that would also be a sort of GMP, of course. And that means that this facility doesn't necessarily have to have a manufacturing license, which you usually require, of course, for manufacturing a drug.
I'd like to raise a thought. Would this person who does the inspection need some specific expertise?
Yeah, I mean, an audit must be carried out by a qualified person or a person under her command, to say. So it's under the the qualified person must do the audit. We usually call it audit if it's from a qualified person, and an inspection if it's from a regulatory authority. I suppose the word mock inspection refers to some sort of audit.
Right. I think so.
Yeah.
Winfred, did you do anything like that in Uganda?
So in Uganda, like I shared, this is something new that we're doing for the very first time. So we didn't do anything like that. So the trial was approved, but we've not done anything like that. So that's why I mentioned that these guidelines are handy, and they will indeed guide the regulators, but also the national ethics committees and other bodies that oversee challenge studies. And I also indicated that our regulatory body, the drug agency, under its mandate, challenge studies are not considered as a clinical, or they're not considered under their mandate. So they look at a drug specifically, not a medicinal product. So this is left to the National Ethics Committee to take oversight of all challenge studies. So we didn't do this. So this guideline is really handy, and it will be helpful as we oversee such studies that come into the country. Thank you.
I still think that it is also important, because when you do something for the first time, you're sort of naive for it and you ask other questions. So it would also be good if you would contribute maybe some of the aspects that were difficult for you, and maybe also provide your comments to the document.
Okay, I'll do that.
Are there any further thoughts? Any further questions, Joris?
I think when I look at the list, sorry, I go through it again. We covered Martin Schouten. Yes, he has a remark on the manufacturing and complying with 21 CFR part 640 was for blood, etc. So, but I think it's in relation with comparison with other regulatory authorizations. So on the white paper, so the quality parameters release, but I think it has been covered for most of them. Adrian, we answered his question. And I think a question about the bioburden from Martin Schouten, a word on that?
I didn't read the question.
So the question is, regulatory authorities have difficulty in judging aspects where challenge agents differ from attenuated vaccines. We already treated that. And for example, then he raises the question to show the bioburden to USP with, for example, where he gives the example of Shigella 10 to the 9th in your product. So would it be prudent to discuss these issues in the white paper? I think we can. I would leave that to the people who write the white paper and discuss that quietly among themselves.
Yeah, and just more generally, if I may, just in terms of the, I think the intent is to actually try to pull out conceptual principles where you have, for want of a better word, challenging scenarios, let's call it, when you're going through the manufacturing of different pathogens, you have different problems, and obviously engagement with different persons manufacturing these challenge agents. This is an ongoing process, so definitely capturing some of those examples and illustrating that in the example section, I think we've recognized is quite a useful element, but we can't go into every potential scenario here.
I think for the sake of time and the respect of the time zones, we came to the end. Peter and Isabel, do you have a final word?
Yes, I would like to thank you all for this very interesting and vivid discussion. We raised a lot of issues. I think you're going to receive some more comments from some people to the paper, and thanks all for the discussion and your presentations.
Thank you, Isabel, for guiding this discussion. I would like to put your attention to the review. We have a few questions from Wellcome Trust and our sponsors. I don't know how we are going to do this. Abby, are you? Oh, it's over there. So it's now on your screen. So please answer to this in now. It is quite simple, and we can do it now. Okay.
Yeah, it's launched, Peter. It's launched.
That's launched. So
I see it on my screen.
To finish, I like to thank everybody, all the speakers, all those who posed questions, and particularly I like to thank the team around Peter, Maddie, Abby, and particularly also Isabelle and Jean-Hugues, who invested a lot of time with people in this very interesting and specific webinar. And now it's time to close, I think. Thank you very much. And please.
Thank you.